Hello everyone, my name is Sunil Nadar. I am one of the PhD student at the CHISH lab at the Microbiology and Cell Science. In this journal club, I will be presenting one of the papers, which is the hyperlink. You can find this in detail in the journal club pod. So the title of this paper is Exposure to Environmental Pollutants Selects for the Xenobiotic Degrading Functions in the Human Gut Microbiome. And this paper is published pretty much this year, and the study was conducted over a couple of years. And this is actually the whole entire research team. It's like there are like more than 10 people associated with this. The primary lead researcher for this team is a professor at the Agriculture University in Italy. That's where the study was conducted in collaboration with multiple other departments. So primary, it's an agricultural university in Italy, and this is her. Her name is Francesca, and then she's the team lead, and she's the one who pretty much the first author and the team lead researcher in this particular study. And this is the department with pretty much the College of Agriculture. So the reference is here, Department of Agriculture Sciences. So why this particular paper is important, and why did I pick this, and why should we definitely care about this particular paper? So to introduce that particular concept, so I'm actually using one of a different a diagram or the different research from another paper. So everybody's aware about our environment has a lot of pollutants, and it has been going through tons of, because of the human activity. We have landfills and air pollution and other different modes of water pollution. So the pollution is one of the biggest common modern challenges that we have. And because of the pollution, so one of the things that we have is the microplastics. And this microplastics, and people just don't even care, and then we just throw it, and then through the food chain, it enter into the small animals, and through biomagnification, and this accumulation of microplastics leads to the upper level, different higher level trophic level of organisms, and then enter in the food chain, and preliminary, that's how it enter into the different organisms. So because microplastics are one of the challenges, and how exactly this microplastics, which is one of the component and is affecting the gut microbiome. So there was a study designed and conducted. So what they did was they picked two healthy volunteers, and they created a design. So that we cannot just give them microplastics, but what they did was they measured the microbiota, which is from the fecal inoculum, over around like 5 days. These are normal, healthy, control individuals. So then what they did was, and they designed the ascending transverse and the descending colon of these individuals in a model called SIMG. So they created this model, and then they stabilized this control microbiota, and then they introduced this microplastics, and then they measure the data for the next 72 hours. So it's very interesting and how the data came out. And so pretty much this is the control data, which is the normal microbiome without microplastics. And this data collected from here, this is actually from with the microplastics. So the data is very, very, very interesting, and the study is actually hyperlinked here. It's you can read that later. So I would like to quickly go over those graphs. And what you notice here is a significant difference in the changes in the gut microbiome. So what you see, the first part is all the control, and then second part, you can see that there's these the three lines are actually ascending colon, transverse colon, and the descending colon of that person. So you can see the gut microbiome is significantly declining. That includes anaerobes and aerobes and staph and enterobacteria. All these species are changing just because of one factor, which is the microplastics. And So, and the same thing that you see here in the second part, there's a decline in with other species. There's a decline in the second volunteer. This is the a second person. It simulates the second person. So they study pretty much the two healthy volunteers, and you can see there's a significant change in the gut microbiome. So this is one of the study. I mean, I thought of picking this graph because it's a very elaborated study connecting to our study, which I'm going to talk about in a few minutes. So microplastics can significantly influence the human gut microbiome. So that's what this study is talking about. So I hope that answered the question and why should we care, and then how this we need to study these environmental pollutants can have a significant impact in our human gut microbiome. The next, I would like to quickly go over some of the key terminology that we use in the study. The first one is xenobiotic. Xenobiotic means any chemical that is foreign to animal life. That could be a pollutant or any type of pollutant that we are talking about. And the next one is the shotgun metagenomics. And this is one of the approach for the sequencing that they use. So entire microbial communities, that's what they used in here. The number three right here is called dioxin, and dioxin is one of the major pollutants that they studied in this paper. It is highly toxic environmental pollutant. The next term that we're going to talk about here is the hologenome. Hologenome is the entire genome that it combining the human and the microbiota. And then operational taxonomic unit, or OTU, that we used here in the analysis part. And the next word is the exposome. Exposome is pretty much, it's a concept that measure all the exposure of a person that experiencing from the birth all the way to the death. So anything that person is exposed to is pretty much, we call it as exposome. And the next one is the PCBs, and PCBs is another another pollutant that we studied in this paper, which is called polychlorinated biphenyl. It's like a group, it has multiple chemicals included in that category. The next one is MAG. MAG means the metagenome assembled genome. So I know that everybody's familiar with this terminology. So these are some of the terminologies that we are using in this particular study. So the study background is actually correlating three different factors. Pretty much the two factors, and the third one is the linking factor. So the first factor is the human gut microbiome health, and the second factor that we are trying to study is the environmental pollutants. So the pollutants, exposure, gut microbiota. So this is how this whole entire study is designed on. So now let's talk a little bit about the pollutants and what pollutants that we studied in this particular paper. So there's like tons of different categories of pollutants that they study. Pretty much the primary group is the dioxins or dioxin-like compounds. That includes different PCDs or polychlorinated dibenzoparadioxins. Sorry, that's a long word. And then PCDFs and PCBs, those are the group of dioxins. They are highly toxic pollutants, which usually comes from water through the water pollution. And the another thing that they studied was heavy metal. Heavy metal, they studied pretty much more than 20 different type of heavy metal. So I just picked one here. I cannot put all the 20 here, sorry. Mercury is one of the example. They used antimony, cadmium, lead, arsenic, all those heavy metals they studied. Another pollutants category that they study was organic pollutants, which that includes the bisphenols and phthalates and polycyclic aromatic hydrocarbons. So all those type of pretty much those type of group that in the perfumes and all that kind of chemicals. The last group that they studied was the other specific compounds that includes PET, which is polyethylene terephthalate, and benzoate, which is food additives. So these are the environmental pollutants, primarily that they focused in this study. The second one is the exposure, how people are exposed to these environmental pollutants, and it's primarily through the ingestion. They can eat or drink and consume in any other form, and then inhaling if it is an airborne particles or particulates or any other form. So they can inhale it, or skin contact through many other form, if they are touching, so these pollutants can get into their body. And then also indirect metabolism. Indirect metabolism means like if a person is consuming a one particular environmental pollutant, and they consume it, it goes to their liver, and the liver metabolites it, they break it down, and so the metabolic products are actually more harmful than the primary product. So primary pollutant, sorry. So that's called indirect metabolism. They just get into the metabolism and then produce a different compound. And the third component here is the gut microbiota. So due to exposure, and the gut microbiota changes, and how it changes we're going to talk about quickly. And these are the groups of gut microbiota that they've preliminary focused on: actinomycetota and pseudomonadota and bacteriodota. So these are the groups which they mentioned in here in the study. So next, we're going to talk quickly about why this study is about. So there are three major purpose that you can elicit from this paper. The first one is how environmental pollution influenced the composition and potential activity of the human gut microbiome. The second one, changes in the gut microbiome upon exposure to the various levels of environmental pollution, how it changes. The third one is the changes in the microbial genes related to the degradation and resistance to the dioxin and heavy metal. So these are the three different purpose or the need that they showed in the study. Now coming to the methodology part, methodology is pretty much simple, but it is very expensive. The experimental design is simple, but it's very, very extensive. So the first one is the study population and the sample collection. So they pretty much screened around 359 healthy adults from the Campania region of the southern Italy. So they were selected based on the residence areas in the different levels of environmental pollution. So they used a pollution index to group these people where they belong to. They are high polluted area, medium polluted area, or the low polluted area. They use a model to distinguish these two. And also they use an inclusion criteria. They just want to make sure that these people are healthy adult, aged between 20 to 50, and both genders, without severe any chronic infection. Also, they are free from HIV, HCV, and HPV. No drug use or no alcohol abuse, not obese, and lived in that area for at least 5 years. So those are the categories that they use. And the samples they collected there are two types of samples they collected. One is the peripheral blood samples. That's to check their serum plasma components, and also the fecal samples. The next part of the methodology is the analysis of the environmental pollutants, which was not that easy. So the study measured pretty much around like 17 different dioxins and PCBs, and for that they used a modified analytical method to study and analyze the trace elements or the heavy elements. So they particularly focused on 19 metals and metalloids in the blood serum using an inductively coupled plasma mass spectrometry. That's the tool that they use to measure the presence or absence or quantify these metals. The third one is the gut metagenomic sequencing and data analysis. That's a very, very extensive part. They collected very large amount of data. So the first step was the DNA extraction and sequencing. So they extracted from the fecal sample using the standard protocol, and then they used the shotgun metagenomes were sequenced all on the Illumina NovaSeq platform. And the second part in is the data analysis. They used the bioinformatic analysis, which is for quality control. They used the Thermomatic Fast QC to remove all the noise. or the low quality sequences. Metagenome assembly and annotation. For that, they used all these tools like a megahit, metabat, and concord, and so on. And then the taxonomy classification to classify these organisms. So they used the NCBI RefSeq genome taxonomy database. So those are some of the tools that they use to classify. And then the fourth part is the functional annotation of the gene. They use the database such as KEGG and COG and a BACMET database to for the metal resistance gene and to identify those particular genes in that genome. You will see that later in the study. So these are some of the tools that they use for the metagenomic, the genomic analysis. For the statistics part, so they use the diversity and composition analysis. They use like a QUIME and also the R packages and Permanova to use the difference in the microbial composition across the different pollution groups, and differential abundance testing, Wilcoxon test, and then machine learning and classification. So SPLSDA, I'll show you that in the graph, and also random forest classification. Then they use the correlation analysis. Also for the correlation analysis, they use the MMVEC, and for the graphs and everything are in the Python package and R. So that's what they use to create the graphs. So the next we're gonna talk about the main part of this paper, which is the result. So the first thing is the first graph that you see here is actually a sparse partial least squares discriminant analysis, which is SPLSDA model. So based on the microbiome taxonomic composition. So what you see here is the gut microbiome composition of the subjects from the different population levels are significantly distinct. So what do you see here? The red circle that indicates the microbiome data from the people who live in the high polluted area, and the green is the low polluted area, and the blue is the medium polluted area. So this model, they use this model to separate these three microbiome data, and this clearly shows that the environmental pollution has a significant difference in the microbial community in the human gut of these individuals. So the clustering of this high and low pollution groups suggests a clear impact of pollution on the gut microbiome composition. So that's the first graph that they showed right here. The second thing that we're going to talk about is a violin plot. And on y-axis right here is the Jacquard's distance based on the gut microbiome composition between the groups. So what you see here is the Jacquard distance means, which is on the y-axis here. So that is the measure of dissimilarity between the sample set. So what they did was they pretty much compare their genome, the gut microbiome genome, within the group and also between the group. So and then they measure how different they are. So if they have more dissimilar, you see the peaks more higher. So the Jacquard distance means it's how different they are. So that's why you see here high and high, which is the microbiome from high area, people from the highly polluted area with the highly polluted area, internal comparison, you see a lower score. So the gut microbiome composition varies significantly between the subjects from the pollution exposure level. From if you see here from medium to low, you see that there's the highest difference or the distance between those groups. And within the group, you see that's smaller difference. So that's what this study, this graph explains. So the next is what do you see here? It's a continuation from the same study. So what do you see here is the box plot. It's the same panel of diagrams as C and D. So this box plot showing the relative abundance of actinomycetoda and pseudo Pseudomonadoda. So those two groups, that's what you see here in y-axis. So they compared this particular group of organism that in the highly polluted area, medium, and the low polluted area in both groups. So one see here, it's the highly polluted areas have the higher relative abundance of actinomycetoda in the gut microbiome, those from the medium and the low polluted areas. And this one, if you look at it, it contrasts the result and revealing the subjects from the medium pollution areas exhibit notably higher relative abundance of Pseudomonadoda group of organisms compared to the low and high pollution area. So this finding pretty much suggests that the different level of environmental pollution selectively enrich specific microbial taxa in the gut microbiome. So the next group, I know that it is a little bit difficult to see these writing here. That's another problem with. So now we are jumping into the pollutants. So the top diagram 2A, this one, it is actually demonstrate the subjects from high polluted areas have a greater abundance of genes related to the degradation of dioxin in their gut microbiome. So in simple language, what exactly it is, so they compared the different abundance of the genes related to the degradation of dioxin. Dioxin is one of the pollutant. So as you see here, if there is more dioxin if they are exposed to more dioxin, and they actually noted there is more dioxin degrading genes in the microorganism live in their gut. So there is when they are exposed to more pollutants, they have a kind of these microbiome is actually changing in their, or I would say they are selected favorably to have more genes to degrade or to break down those pollutants. So which is pretty much good thing, but we still don't know how the pathogen, how this is really affecting the people. And the figure 2B is pretty much the same as well and shows these same subjects have higher serum concentration of various dioxin classes. So this validate in how much dioxin they have in their blood, and this graph shows that how much protective genes or the breaking down genes that they have in their gut microbiome. So this finding actually suggests that the exposure to the high level of environmental pollution, particularly dioxin, leads to the selection and enrichment of microbial genes capable of degrading these pollutants in the human gut microbiome. So quickly going through a few more diagrams, and here this one, what do you notice here is this demonstrates the subjects from the medium pollutant groups have more genes related to the heavy metal resistance. So this is the same thing, but it is related to the heavy metal exposure, and it selects the antibiotic resistance gene. So that's what you see here. The next is, I'm going to quickly go through this because of the time, and this part right here is also shows that the same subject also have a higher abundance of antibiotic resistance genes. So this one is the heavy metal resistance genes. So, oops, sorry. So the next graph is actually showing the heavy metal concentration is higher in the subject from the medium environmental pressure area. So this is actually a little bit, this data is a little bit different from what they were expecting. So what do you notice here is the subjects from the medium environmental pollution area exhibit the higher plasmatic concentration of various heavy metals compared to those from the high and low polluted areas. So this graph in figure 5A is actually showing that the resistance genes are widespread in the microbial genomes reconstructed from the metagenomic reads. So here it shows that there is a significant proportion of MAG, particularly those from actinomycetoda, contains the gene for dioxin degradation. And 5B, this one is showing that the heavy metal resistance genes are widespread across different bacterial taxa that you see here on the right, with a higher prevalence in the medium pollution group. And the last one right here, 5C, indicates the antimicrobial resistance genes are also widely distributed, particularly in the medium pollution group. So this is an very interesting graph. They try to correlate between the heavy metal resistance gene and antimicrobial resistance gene. So, and prove that they pretty much co-evolve these two things. So the bar chart here indicates that the co-selection of these genes in prevalent in the specific microbial taxa that you see right here, with the Pseudomonadota and Bacillota being the most represented. So these findings highlight the impact of environmental pollution on the gut microbiome, driving the co-selection of these resistance genes and potentially contributing to the spread of antibiotic resistance. That's not good. So coming to the last end of this paper, there are four main important ideas that they discuss in the last part of this paper. The gut microbiome can adapt to the environmental pollution. High environmental pollution can significantly change the composition and function of the gut microbiome. The study also indicates the environmental pollutants drive the co-selection of heavy metal resistance gene and antibiotic resistance genes. The unexpected result was actually in the higher plasma concentration of heavy metals in the subjects from medium pollution areas compared to the high pollution areas. Coming to the conclusion, there are three conclusions that we can make out from this paper. The environmental pollutants have significantly influenced the human gut microbiome, driving the selection of microbial functions that degrade xenobiotics and resist heavy metals and antibiotics. These adaptive responses indicate the microbiome's resilience and its crucial role in mediating the effects of environmental stressors on human health. The research also highlights the need for comprehensive environmental health strategies considering the gut microbiome's role in mitigating pollution-related health risk. So a couple of questions that we can think of related to this paper maybe for future study. What would be the long-term health effects because of these changes in the gut microbiome due to this chronic exposure to environmental pollutants? And what specific genetic and metabolic mechanism enabled the gut microbiome to adapt and to degrade the environmental pollution? So you have noticed that they particularly look for the dioxin degrading genes, and which was more abundant when they are exposed to a high amount of dioxins, and which they proved it from the serum plasma concentration. So what would be that specific metabolic mechanisms enable that? That would be a cool thing to study. And another thing is, how does the horizontal gene transfer contribute to spreading of the xenobiotic degrading and resistance gene within the gut microbiome under environmental pollution pressures? And you know that the horizontal gene transfer that happens between the different microorganisms. So at what level these genes are going to be transferred from one to another? And the last one we can talk about as can this composition and functional capacity of the gut microbiome can be used as a bioindicator of assessing environmental pollution levels and associated health risks. Probably like we can find out, okay, what was this person's geographic distribution after testing this gut microbiome, how long they lived, and it's something like counting the growth rings in the the tree trunk. To find out the years, and maybe we can use it as an indicator, and somebody has to validate that probably later after multiple repeats. So these are some of the interesting that maybe we can study as a follow-up, and here are some of the references that I used, and that's all about this paper, and I love to see the discussions, and hope you enjoy this, and please let us know your feedbacks, and we will talk about that in the discussion form. Thanks so much. I appreciate you listening.